At the site of an injury, your innate immune response is immediate. Immune cells such as dendritic cells and follicular dendritic cells are able to detect pathogens, such as extracellular bacteria in this case, entering your body in different ways. Here we are about to see a dendritic cell interact and phagocytose a pathogen via receptor-mediated endocytosis, the pathogen that it encounters from the site of infection. Dendritic cells have many TLRs, which recognize specific pathogens. This causes them to change gene expression and start migrating into secondary lymph tissue. By the time they get there, they no longer take up antigens anymore. They become mature, and they focus on presenting antigens to naive T cells. Oh, paper cut.
Hi, I'm a dendritic cell. I hang out in the tissues most of the time, waiting for a pathogen to phagocytose. When I do, I take out my handy-dandy cytokine, IL-15, and I recruit neutrophils to the site of the infection. What's this? This looks like a pathogen. Ooh. That means release. Where's IL-15s? Cookie! Yeah.
After phagocytosis, the dendritic cell becomes mature or activated and grows finger-like processes called dendrites, so it can better interact with T cells in secondary lymph tissue. You're about to see what is happening inside the dendritic cell. The antigenic peptides are being transferred from the cytosol to the endoplasmic reticulum, also known as the ER. Transport of the peptides across the endoplasmic reticulum membrane is accomplished by the transporter-associated antigen processing TAP. The MHC receives the peptide from TAP1 and TAP2 to then present it to the antigen-specific T cell.
All right, doctors, tap 1 and tap 2. We're gonna take this. All right, let's try and cut this pathogen up.
Hurry.
We gotta try and present it to MHC class 2.
We need to present this peptide. Present it.
Present it in a present. Red piece, red piece. There you go.
Yeah, you're right. That was the wrong one.
All right.
Now I got
Red MHC class 2.
I got some MHC class 2 ready to present the peptide. In. Ready. Okay.
Dendritic cell, back again. I'm mature and I'm hanging out in the secondary lymph tissue, not like the immature dendritic cells that are out in the tissues still. Along with MHC class 2, I'm going to bind to a naive T cell with B7 that was put on my surface when I sensed that there was inflammation and an infection. This is why vaccines need to have adjuvants so that they can put B7 on my surface and activate the adaptive immune system. I also have ICAM, which is going to strengthen and prolong the bond between me and the naive T cell.
Hi, naive T cell here. Got my handy dandy TCR along with the CD4 to help recognize the MHC class 2 molecule. Over here, got my CD28 that interacts with the B7 on my dendritic cell partner, as well as LFA1 that interacts with the ICAM on the dendritic cell that helps prolong the interaction so that I can become activated.
MHC class 2 binding with CD4 and the TCR. Then we got ICAM and LFA1 and B7 and CD28 all bound together.
Oh! Activated! Oh, what are these? IL-2s? I just feel like differentiating and proliferating.
Now that the T cell is activated, it can proliferate and differentiate into different types of effector T cells. Cytokines are key in determining which type of effector T cells will be made. There are Th1, Th17, Th2, TFH cells, and T regulatory cells. All of these will help aid and suppress in the immune response. Join us next time when clumsy Maggie gets a paper cut or another open wound, and we have to redo this again. Now that the T cell is activated, Now that the T cell is activated, it can differentiate into many different types of effector cells. Cytokines are very key in determining...
You're shaking the paper!
I told you, I get nervous! Oh, sorry, I was looking down, I didn't...
Wait.
Tell me when to go with that.
Okay.
Sorry.
No, you're good. No, that was... Okay, stop! Oh.
Go.
At the site of an injury, your innate immune response is immediate. Immune cells such as dendritic cells and follicular dendritic cells are able to detect pathogens such as extracellular bacteria in this case. Pathogens such as Mother of God!
Now that the T cell is activated... Why is that so funny?